In this lecture, we are discussing Chapter 6, the neurotransmitter control of hypothalamic, pituitary, and other hormones. This chapter deals with how neurotransmitters influence the release of hypothalamic and pituitary hormones and the hormones of the adrenal medulla, pancreas, thymus, and GI tract. These we will not discuss in any great detail at this time. And with the effects of neurotransmitters on the neurosecretory cells, again, in an introductory and overview fashion. Granted, a high degree of memorization in this exercise, but this is our method and approach to developing our general understanding of neuroendocrinology and appreciation of the complexity and the diversity of the functional mechanisms involved. This figure is an overview of that, as stated, focusing on the hypothalamo-pituitary gonadal, or HPG axis. We see that somewhere in the nervous system there is a nerve cell with release of a neurotransmitter. The neurotransduction, neuroendocrine transducer role is key at this point. In this case, the neurotransmitter we're considering is noradrenaline, and it is targeting a hypothalamic neurosecretory cell, in this case the GnRH neuron, stimulating the synthesis and release of GnRH, so increased action potentials coupled to the exocytotic release of noradrenaline increases the firing rate of the neuroendocrine cell, stimulating synthesis and secretion of GnRH, actually affecting the pulsatile release, as we'll develop a concept that we will develop further as we proceed in this discussion and future discussions on the HPG. Again, through the hypothalamo-pituitary portal system, the vessels, delivery through these portal vessels, I don't like to use the term veins, to the target cells, the gonadotropes within the anterior pituitary, mediated via transmembrane signal transduction through a second messenger system. The second messenger system, as you will see, cyclic AMP, stimulates both synthesis at DNA and then RNA, and secretion, exocytosis. And the gonadotropins, LH and FSH, are released into the general circulation, and the bloodstream ultimately delivers it to the target cells within the target organs, the gonads. Again, transmembrane signal transduction, receptor-mediated activation of a second messenger system. We'll see that for both of these gonadotropins, cyclic AMP is also the second messenger, and this activates a series of enzymes triggering the synthesis of the hormones 3 for the gonads, in this case estrogen and progesterone, obviously in the female. And as soon as they are synthesized, they diffuse out of the cell and are washed away by the plasma, and delivered ultimately through the systemic circulation, the bloodstream, to the target cells, acting on the receptors at the level of the genome to impact protein synthesis, for example, in an endometrial cell. So this is the general mechanism. Of course, what was not shown in the prior slide was mechanisms for negative feedback that we'll discuss in later chapters to complete the regulatory circuit. But at this time we're thinking of the drive to the hypothalamic neurons that are either projecting to the posterior pituitary or to the anterior pituitary. The example that we see in this slide again is the noradrenaline neuron, noradrenergic neuron, with neuroendocrine transduction onto a hypophysiotrophic neuron with its terminal ending on the primary plexus, first capillary bed of the hypothalamo-pituitary or hypophyseal portal vessel system, and the hypothalamic hormone releasing or inhibiting hormone will be delivered to the anterior pituitary, as we understand. So what we have to picture are all of these neurons that are involved in hormone release either at the anterior pituitary or posterior pituitary, including the tuberoinfundibular dopaminergic neurons shown here, have tens of thousands of synaptic input onto them and are integrating all of that excitatory and inhibitory neurotransmitter and neuromodulatory effects by all of the neuroregulators collectively impinging upon them and converting that into a firing rate, determining the rate of exocytosis and release of the hormones. So the neural inputs to the endocrine hypothalamus, there are many of these. For example, acetylcholine, dopamine, norepinephrine, NA is noradrenaline shown here. We should change all the NAs to norepinephrine, the standard term now used for that transmitter, that chemical messenger, let's call it. Serotonin, and of course these are acting in the hypothalamus and stimulate receptors on the hypothalamic neurosecretory cells. We need to add to this list, of course, glutamate and GABA, the major gas pedal and brake, respectively, or excitatory and inhibitory neurotransmitters that act rapidly to depolarize or hyperpolarize, again, respectively, the neuroendocrine cells of the hypothalamus. So the secretion of the hypothalamic releasing and inhibiting hormones through their action on the synaptic receptors of the neurosecretory cells is regulated by different neurotransmitters from nerve cell axons. No surprise there. The neurotransmitters can act in 4 different mechanisms to regulate hormone release from the hypothalamus and the pituitary gland. In the neural pathways, the first of these in the neural pathway with sensory receptors input, between axons from extra-hypothalamic neurons and neurosecretory cells, from nerve cells having synapses on the axons or nerve terminals of neurosecretory cells, and finally released into the hypophyseal portal veins, having an effect there. This figure is showing that composite of the 4 mechanisms. So first, indirect through an interneuron. So here we see an extra-hypothalamic neuron projecting to a neuron that might be either, in this case, they're showing, in this example, they're showing noradrenaline, so it would be extra-hypothalamic. This could be sensory information to the locus ceruleus with the projection of the locus ceruleus noradrenergic neuron onto a neuroendocrine cell, secretory cell. Second example is a direct axodendritic or axosomatic synapse on the secretory cell. So we see that's true in both of these cases. Here we're seeing another neurotransmitter, serotonin, acting in a neurocrine fashion across the synapse. Here targeting the postsynaptic receptors on the hypothalamic neurosecretory cell. Third, there can be axonal synapses on the neurosecretory cells, shown here in the median eminence, where the presynaptic neuron can alter the membrane potential of the axon terminal, either enhancing the release with action potentials arriving or blunting action potentials by hyperpolarizing the membrane and decreasing release. And then finally, number 4, the release of a transmitter directly into the portal plasma and delivering it to the anterior pituitary to act directly on the target cells in the anterior pituitary. Then the neuroregulation of the adenohypophyseal hormones is what we will be viewing in the subsequent slides. There will be a separate lecture recorded on this research article by Itoy et al., Differential Regulation of CRH and Vasopressin Gene Expression in the Hypothalamus by Norepinephrine, from the Journal of Neuroscience, Volume 19, Issue 13, pages 5464 through 5472, in the year 1999. So that will be a separate lecture as an example of primary literature dealing with at least one of these axes, the hypothalamal-pituitary adrenal axis. Then some of the information from our textbook reporting CRH and ACTH being stimulated by serotonin, dopamine, norepinephrine, and epinephrine inhibiting CRH release, but these can be excitatory, as we will see, during stress, the norepi and epi, along with the ACTH, can increase the release of ACTH, stimulates ACTH, not along with ACTH. I was reading this as acetylcholine. Sorry, don't do that. ACH increases the acetylcholine now increases the basal and the stress-induced release of ACTH by acting on the CRH neurons. And GABA, the brake, the fast-acting inhibitory neurotransmitter, inhibits the stress-induced CRH and thereby ACTH release. So we have examples of gas pedals and brakes, neurotransmitters and neuromodulators, and of course there is evidence of glutamatergic input to CRH neurons that contributes to postsynaptic excitation or depolarization of the membrane. And then if any of these others are acting through second messenger systems to be neuromodulators, they can all work together to affect the firing rate of the CRH neurons, or the firing patterns. So we'll just look at each of the illustrations from the textbooks for the different neuroendocrine axes and go by the author's statements of the effects of various neurotransmitters as far as any exam questions would be concerned. Therefore, memorization of these effects on the neurosecretory cells of the hypothalamus is key. So stress and circadian rhythm information from the suprachiasmatic nucleus mediated via serotonin can activate or enhance firing rate and release, and therefore synthesis of CRH. Norepi and epi and dopamine are shown to inhibit here, and as I said, we'll challenge this idea of norepi being inhibitory in that research article that we had mentioned. This reminds us that many of these effects are state dependent. So a little bit beyond memorization and understanding is the key here. The acetylcholine as a gas pedal, as we said, especially during stress. GABA as a major brake, inhibitory neurotransmitter. And then, of course, the CRH stimulatory to synthesis and secretion of ACTH. Vasopresin also can act as a secretagogue to ACTH, and we've seen some examples, and we'll see additional ones in the research article that I had mentioned on the vasopressin. Indeed, CRH neurons can be induced to express the vasopressin gene, and do express it constitutively in many cases, and there are different times when the CRH is functioning as the secretagogue and when vasopressin is. And we saw this already in the example of the paper on the interaction of the HPA and the HPG axis. Then the corticosteroids, of course, are ultimately released, and they produce their negative feedback at the level of the hypothalamus and the pituitary for negative feedback. The adrenal medulla would be activated during stress, and the norepi and epi that are released through the sympathetic nervous system can have a positive effect at the level of the anterior pituitary on ACTH release. And in the previous slide, this was the stimulatory effect of these hormones now, not neurotransmitters because they're not acting synaptically, on the ACTH, the corticotropes directly. For the HPT axis, the effects on the release of thyroid-stimulating hormone from the anterior pituitary, dopamine reduces baseline TSH levels and inhibits cold-induced release of TSH by inhibiting the release of thyrotropin-releasing hormone, or TRH, from the hypothalamus, and by inhibition of tryptophan cells in the pituitary. Epinephrine and norepinephrine stimulate TRH and TSH release, similar to what we had seen in the model for the CRH, in that they act both at the hypothalamus and at the anterior pituitary. So both stress and cold can be the stimuli coming in. Stress will excite or enhance acetylcholinergic neuron activity, increasing the gas pedal or the drive to the TRH neurons. GABA, of course, is a major brake on this. Stress will inhibit through mechanisms not indicated here. The neurotransmitter is not identified by the authors. Cold also activating norepi and epi, and those transsynaptically will activate TRH neurons. Dopamine inhibiting, and serotonin having both positive and negative effects on these, indicating neuromodulatory role there. The stress will also activate somatostatin release that once it reaches the anteropituitary has an inhibitory effect, and of course anything that will increase the release of TRH will drive the gas pedal for synthesis and secretion of TSH. We see that blood-borne dopamine can have an inhibitory effect directly on the thyrotropes in the anterior pituitary, and of course the thyroid hormones, so thyroxine and triiodothyronine, have negative feedbacks on both the anterior pituitary thyrotropes and the TRH neurons themselves. For the HPG axis, when we consider LH and FSH release from the anterior pituitary, of course we know we are talking about hypothalamic pituitary gonadal axis. So the GnRH neurons will be the targets of the neurotransmitters, and the gonadotropes FSH and LH will be the anterior pituitary's hormones, the gonadotropins that we consider. This slide is illustrating part of the menstrual cycle that we'll discuss in detail later, but we see here that it is a very complex series of events that occur over approximately a 28-day time point, and thereby many different things happen. We can have stimulation and inhibition, so in other words negative feedback and positive feedback, so many things change, and that's why we end the semester focusing on the HPG axis to look at these complexities. So the effects of neurotransmitters on GnRH are what we're considering now, and it is of course going to be complex. There are two hypothalamic areas of GnRH neurons, the preoptic area and the mediobasal hypothalamus, and both are going to be responsible for GnRH release, that is the drive for the synthesis and secretion of the gonadotropins at the level of the anterior pituitary. So we see first that acetylcholine is negative inhibitory on both of these. Serotonin is inhibitory on the mediobasal hypothalamus, but is excitatory as being driven from the suprachiasmatic nucleus on the preoptic area GnRH neurons. Dopamine has a stimulatory, excitatory effect on the preoptic area neurons, and when estrogen levels increase in the female, help contributing to the switch to the positive feedback, they have an excitatory effect on the estrogen has an excitatory effect on the preoptic area GnRH neurons, part of which is mediated via the dopamine neurons. This will be important when we talk about the LH surge. As I said, in the female, there is a switch from negative to positive feedback that is mediated obviously by the high levels of estrogens produced at a certain point in the menstrual cycle. The GABA at the mediobasal hypothalamic GnRH neurons is of course inhibitory, and then we have already looked at how the opiates inhibit noradrenaline, which is a gas pedal, to the mediobasal hypothalamic GnRH neurons. And the gonadal steroids have an effect here as well. And exogenous opiates can act here on the noradinergic neurons and decrease GnRH, inhibiting the HPG axis altogether. And then, of course, the gonadotropins stimulate estrogen and progesterone and testosterone synthesis and secretion, each of which has negative feedback on the different levels. And the estrogen, of course, has a role in switching from negative to positive feedback. Our focus now is on the neurotransmitters, not so much on the axes themselves. So please stay focused just on that, and you're preparing for an exam. Prolactin then, dopamine is the main prolactin inhibitor, as we know. Adrenaline and noradrenaline, or epinephrine and norepinephrine, inhibit the release of prolactin. Serotonin might stimulate prolactin release by the inhibition of dopamine secretion. Acetylcholine might inhibit the release of prolactin, maybe by elevating dopamine, and GABA stimulates prolactin secretion by inhibiting the dopamine release. We see that many of the mechanisms for increasing prolactin release is through disinhibition. You inhibit an inhibitor and you disinhibit the prolactin synthesis and secretion by the lactotropes. So here we see this in the illustration. Sensory input, either cervical stimulation or suckling, and of course stress, all have a gas pedal-like effect or excite serotonin neurons, increasing the release, and the serotonin will excite prolactin-releasing hormone, possibly VIP, and that will drive the release of prolactin. As well, the serotonin can inhibit the prolactin inhibiting hormone secretion, inhibit the dopamine neurons, so disinhibit prolactin. Norepi and epi have seemingly opposing effects sometimes, opposite effects sometimes, so this is that neuromodulatory effect that we would be considering. Under certain situation might be excitatory, under others might be inhibitory, depends on the interaction with other transmitters. Acetylcholine having a direct excitatory effect on the dopamine release, and GABA, obviously the brake pedal, and opiates for the endogenous, opiates refers to exogenous, but anyway, an inhibitory effect on dopamine, so disinhibiting and increasing prolactin release. And then estrogen can have effects on the serotonin, stimulatory effects, as well as stimulatory effects on thyrotropin releasing hormone that also has is another secretagogue of prolactin from the lactotropes. And serotonin can act directly on the lactotropes, increasing prolactin synthesis and secretion, and GABA directly at the level of pituitary, decreasing prolactin synthesis and secretion. Estrogen feedback is a positive drive for prolactin release and for inhibiting, and it has a potent inhibitory effect on the dopamine prolactin inhibiting hormone system. So disinhibiting as well as having a direct effect at the anterior pituitary. And then the estrogen, as we see, can increase the drive of prolactin release through the serotonin system and the TRH system, but the prolactin has a direct negative feedback effect on the TRH. Next, the HPL or HP liver axis for growth hormone is controlled by growth hormone releasing hormone and by somatostatin, so gas pedal and brake. However, neurotransmitters such as dopamine, acetylcholine, and norepinephrine seem to increase GH release. Now we can have a brief discussion of the neurotransmitter regulation of the posterior pituitary, or the neurohypophyseal hormones. So acetylcholine is a primary transmitter for the stimulation of oxytocin and vasopresin release. It is responsible for stimulating oxytocin release in response to suckling during lactation. And dopamine inhibits milk ejection and oxytocin secretion in response to suckling. So we see this for oxytocin shown here. There are many other CNS inputs, but it's primarily the neural inputs from the mammary glands through the suckling and from the reproductive tract that have an excitatory drive on acetylcholinergic neurons that synapse on oxytocin neurons and excite them and stimulate synthesis and secretion. As well, there might be other mediators in the sensory circuitry that drive oxytocin release. Norepi and epi have an inhibitory effect. Dopamine also has an inhibitory effect, so the catecholamines collectively inhibit oxytocin release. And then there is the the positive feedback of the oxytocin onto itself. For the vasopressin or antidiuretic hormone, the increased water reabsorption in the kidney is what will have the negative feedback on the osmoreceptors, so it can be positive or negative. So as plasma or interstitial cell osmolarity increases, that's when you're going to have the drive for the vasopressin, and when it decreases to normal levels, it'll have an inhibitory effect. But there are also many other systems that are involved. Norepinephrine, shown here to have an inhibitory effect on vasopressin. I believe if we looked in the literature as well, there are situations and mechanisms when this is a drive. Dopamine excites vasopressin neurons. Acetylcholine is another major gas pedal, as we had said. Histamine has a vasopressin releasing effect, and angiotensin II also has a vasopressin releasing effect. And of course, the vasopressin is going to increase the water reabsorption to bring plasma and interstitial osmolarity into homeostatic, physiologically appropriate levels. We've had many discussions on the electrophysiology of neurosecretory cells and neurotransmitters in discussions from Chapter 5. We'll try to have a few papers that focus on this as well. We're not going to say much more that after the cell firing, the neurotransmitter is released from the axon terminals to the synapse. Just as well, when a neurosecretory cell fires, it increases the release of neurohormones into the circulation by exocytotic release. So we're not saying much at this time, but we will have a few examples as we proceed on this. This statement here, of course, is a rephrasing, if you will, of the neuroendocrine transducer mechanism, neurotransmitters driving neurosecretion of neurohormones. And then the authors end this chapter, as far as the parts we're going to consider, focusing on the complications in the study of neurotransmitter control of hypothalamic hormone release. And you should familiarize yourself with the discussions of each of these matters in the textbook. And again, we'll try to use examples as we go through the semester to demonstrate some of these challenges and complications. So there are many potential neurotransmitter interactions. There's non-specific drug effects that have to be considered. Certainly in vivo in the whole animal versus in vitro, just hypothalamus or anterior pituitary studied in a separate culture, and the potential drug effects there, ignoring everything else in the entire body versus the whole animal, where it's difficult to get the drug directly to where you want it to have its effects and to assess its contribution to the overall coordinated hormonal responses. There are many species differences. There are sex differences. We already mentioned the female HPG axis obviously being very different than the male. Interaction, but that's not the only example. Interaction with other hormones, anesthetic use, psychological factors, and multiple hypothalamic pituitary interactions. Each of these contributes substantially to the complexity and the challenges of understanding the roles of neurotransmitters in the control of the hypothalamic hormone release and therefore anterior and posterior pituitary function. It is a diverse and dynamic system that is very, very state dependent, which is what we would expect. So this ends our